Hello everyone, welcome back to Biocarrion. I ended my last video with this. I was talking about DNA replication in prokaryotes, and I ended my last video with this point where DNA polymerase 1 and FEN1, the two enzymes which help in removing this these RNA primers and adding up of new DNA. I spoke of DNA initiation and elongation, and in this video I will talk about DNA termination. Now before talking about termination, I will talk about an important question which is always asked by students. Right? The question is, DNA polymerase has exonuclease activity. It can remove the RNA primer from this side. Suppose if I draw it, if I remove this, suppose this was the RNA primer like this red which we had drawn over here in 5' to 3' direction, and along with it this is the DNA with it is attached. Now the question is, which is the actual enzyme which is responsible for removing this RNA primer? Is it DNA polymerase 1 or is it the RNase enzyme? Which of these enzyme is there, or it is the RNase H? RNase H by its name suggests RNase, it removes RNA. And DNA polymerase 1, it also exonuclease, it has exonuclease activity in both directions, in 5' to 3' direction also, and in 3' to 5' direction also. So both these enzymes in combination remove this RNA primer from this side. Experiments have shown that RNase H is found in all species ranging from bacteria to archaea and to eukaryotes. Everywhere this RNase H is found. DNA polymerase 1 is found only in prokaryotes. It is not found in eukaryotes. But since it also have the exonuclease activity, therefore it removes. So none of the important question is neither this DNA polymerase 1 nor RNase H acts alone on this RNA primer. See, suppose RNase H or DNA polymerase 1 starts removing this RNA primer from this side. Now they have 5' to 3' exonuclease activities. Primer is removed from this side. Slowly they may remove the DNA also from here. Now this is not the case. They don't remove the DNA. Why? Why they don't remove the DNA? Question is, right, what happens? Suppose I redraw the DNA over here. What happens? The RNA primer is removed only till this last nucleotide. After the last nucleotide is left, which is the RNA, the last RNase H starts removing this RNA and DNA polymerase starts removing this RNA, ribonucleotides. DNA polymerase 1 in the meanwhile starts adding deoxyribonucleotides in this direction, and this is being removed in this direction, right? And then still DNA polymerase is adding only till the end, till it reaches, and one single nucleotide should be left. In the end, when it reaches here and this RNA becomes DNA, when this RNA becomes DNA, it reaches the end. At this time, one ribonucleotide is left. It forms a flap-like structure that I had written over here. This flap-like structure is removed by the enzyme FEN1, stands for flap, and EN stands for endonuclease, F for flap. This removes this flap. Now if this flap is not formed, then DNA will also be removed by the exonuclease activity of DNA polymerase 1. Right? So the three important enzymes which are responsible for removing this primer from the side, from the sides in lagging strand and also in the leading strand, are RNase H, DNA polymerase 1 and flap endonuclease. All these three are important. Flap endonuclease ends the process of removal of primers. Right? Now next we'll talk about termination. Now for termination, now please note I had drawn this diagram and have not deleted it till now. See, DNA replication, this bubble size is increasing. Now also it looks like a theta. This is an inverted theta. Initially it was a sideways theta. So any worker who works on it and starts reviewing this will always find a theta. Right. Now DNA replication always occurs in clockwise direction. DNA replication in prokaryotes always occurs in the clockwise direction. So helicase, here helicase is present and this helicase is still deloading, removing the helices. So till what point will this helicase remove the points? Will unwind the DNA. Till what point is this helicase going to unwind the DNA? That is the question. Now, for that, see, there are termination sites in bacteria which are known as TERs. And if DNA replication started at suppose this point, suppose this was the ori, so TER sites will be present somewhat 90° past the origin site. So if there was the origin, here the termination sites will be there. There are 7 termination sites in E. coli and these termination sites are known as TER. Right. There are 7 termination sites. 7 termination sites are there and these are known as TER. Now what are the names? See, TER G, TER F, TER B and TER C are 4 termination sites which are present in the counterclockwise direction. And there are 3, TER A, TER D and TER E, TER D and TER A, EDA. 3 sites are there which are present in the clockwise direction. Now I will delete this diagram. First I will draw it here. See how these sites are present. If this was, I will redraw this in the same fashion. So how these are present? See, this is the counterclockwise direction and this one is the clockwise direction. I hope you understand this much. So here TER G is present, here TER F is present, here TER B is present and here TER C is present. G, F, B, C. Go for biology class. You can learn it like this. Go for biology class. So that is G, F, B and C. 4 TER sites. Now these 4 TER sites, they have their directions in this way. In this side, the direction is there for the 4 TER sites. Right? They are just like arrows, the sites are present here. Right? Now 3 TER sites E, D, A are present in the clockwise direction. Clockwise means one is present here, here and here. Right? Now I will name them E, D and A. This is TER E, this is TER D, this is TER A. Right? Now when helicase comes from this side, the replication, the helicase will pass through E, D and A because this is in the same direction. Here a protein is present which is known as TUS. On each of the sides this protein is present and I can draw it in blue. So suppose I draw it like this. This protein is present on each of these sides. Protein is present on each of these sides. Right? So from this side, the E, D, A are in the same direction, but when the helicase reaches this point, either C or B or F or G, just like a railway platform, the rail is slowing down. So it may slow down completely at C or at B or F, and finally it has to slow down, it has to stop at G. So when it reaches this point, this G point, at this point the replication is going to halt, and this helicase will come out of the whole system. The helicase will be totally out. Similarly, now whatever coming from this side, the replication was also opening, this chain was opening from this side also. We learned this before, one helicase is enough. When it starts opening, so this side also starts opening up, the other side also starts opening up, that much is enough. Because if one side is unzipped, the other side will be automatically unzipped in a circular structure. So if in counterclockwise direction, the helicase will encounter G, F, B and C. But when it comes to A, D or E, it has to halt over here. And in this process, this TUS protein which is present over here, it is the loading type of protein, it is the stopper. It stops, it helps these TER sites, it is present at the TER site, which actually stops the overall process. Right, now I will rub this part. Stops the overall process. And one thing that I have missed, you have to learn this part also, eDNA. So I call them EDA protein, EDA protein or EDA site. That is up to you, how do you want to learn it. Now if you come to this part, now in the end what happens, that we have to understand. See, in the end you have formed 2 DNA proteins. Now this is the first strand, and the second strand that has been halted over here. The DNA polymerase, when DNA polymerase has come out of this strand now, so no more DNA polymerase, so no more synthesis, right? And the clamp that was attached with the polymerase, that has also been removed. This helicase has also been removed. Now what happens, in the end 2 strands are there and one inside the other, inside the other, just like one loop present inside the other loop. It is like this, one loop present inside the other loop. At this point of time, what happens, the enzyme is topoisomerase 2. Topoisomerase 2 brings a cut at this position. Right, now this cut will bring the second ring out of the first ring immediately. So one DNA ring will be formed, one DNA chromosome, and the second chromosome will be formed. Right, this is the second chromosome. Now this is not showing you the semi-conservative mode of replication. Now one of them should be red, which is the newly formed strand, right? And the other one should be black in color. This is the full scenario. I'll draw it again. Should have drawn it like this. You should have drawn it like this. This was the original strand, and again this is the original strand, right? And new strands are synthesized on this original strand. So 2 strands are synthesized, and the cut produced by topoisomerase 2 is on topoisomerase 2 is at this point, which will form 2 strands. One is this and other one is this, and the semi-conservative way of replication. Right. Now this is all about DNA replication in prokaryotes. Hope you like this video. Thank you.